Chapter 10, Sensory Physiology. 1 Part 1 goes over sensory physiology, understanding how information is transmitted to the CNS from the peripheral nervous system. There are sensors or receptors in the peripheral nervous system capable of detecting specific sensory signals or sensory modalities and sends those signals to the CNS. Two additional graded potentials to EPSPs and IPSPs are generator potentials and receptor potentials. 3 Pacinian corpuscles detect mechanical stimuli like deep pressure on the surface of the skin. Once the sensory part of the receptor is distorted, induces the axon part of the first-order neuron called afferent or ascending neuron, which is a sensory neuron, to fire action potentials and propagate those APS towards the spinal cord. 4 The sensory neuron or first-order neuron has the cell body located in the dorsal root ganglion. 5 Each ganglion contains hundreds of cell bodies of first-order neurons, which have their peripheral axons wrapped by Pacinian corpuscles. 6 Pacinian corpuscle, when distorted, generates generator potentials that add up to reach the threshold potential to trigger the first-order neuron to fire an action potential. 7 Pacinian corpuscle falls under the one of 4 general senses called pressure. 8 Receptor potential is used by special senses. 9 Microscopic space between a sensory neuron and the peripheral receptor. 10 Stimulus is a chemical from food dissolved in water. If it's not dissolved by water, it cannot be tasted, can be recognized by a taste receptor. Stimulus binds to the receptor and causes depolarization, activating a voltage-sensitive calcium channel, influxes calcium, inducing vesicles containing neurotransmitters to fuse with the cell membrane to empty their contents into the microscopic space. 11 4 General senses, 5 special senses. 12 80% of ability to taste food is dictated by the sense of smell. 13 Certain peripheral receptors are capable of remaining active as long as the stimulus is present, known as slowly adapting receptors or tonic receptors. 14 Specific examples of tonic receptors are muscle stretch receptors and Merkel discs. 15 Muscle stretch receptors detect the length of the muscle, to detect any tears. 16 Merkel's discs detect pressure like touch. 17 Receptor adaptation consists of slowly adapting and rapidly adapting receptors. 18 Rapidly adapting or phasic receptors respond only when there's a change in a signal, like when the stimulus turns on or off. Even when the stimulus is present, it does not respond. 19 Olfactory receptors are phasic receptors. 20 Pacinian corpuscles detecting deep pressure are phasic receptors. 21 Sensory pathways involve 3 sensory neurons in series, one after the other, starting with the first-order neuron, is a sensory neuron belonging to the peripheral nervous system. The cell body is aggregated in the DRG, the dorsal root ganglion. 22 Each dorsal root ganglion consists of hundreds of cell bodies of first-order neurons. 23 First-order neurons fire action potentials towards the spinal cord. They synapse with another sensory neuron in the sensory nuclei of the spinal cord called a second-order neuron, or within the distal posterior gray horn of the spinal cord. 24 Second-order neuron conveys an action potential toward the thalami, where the third-order neuron is located, connecting each thalamus to the cerebral cortex, or the primary somatosensory cortex, or the postcentral gyrus. 25 Primary somatosensory cortex is the functional name of the cerebral cortex. 26 The postcentral gyrus is the anatomical name of the cerebral cortex. 27 The cerebral cortex is where a sensation becomes a perception, or where you become consciously aware of a sensory stimulus. If information fails to reach the primary somatosensory cortex, the individual is oblivious to the existence of that stimulus. 28 Only the first-order neuron is a peripheral neuron, the second and third-order neuron are part of the CNS. 29 Sensory unit is defined as one single first-order neuron and all the receptors it is serving, or the receptors sending information to it. 30 Receptive field is used by 3 specific modalities or sensory pathways, used by tactile or sense of touch. Proprioceptive is receptors in joints and allows knowing body position in space. Sense of sight as well, also called visual fields. 31 Receptive field has a flaw. Distinguishing 2 stimulus from 2 different areas in the same receptive field is impossible, come back as the same stimuli. 32 Action potentials only have one strength. sensory coding allows the brain to tell the strength of different signals. 33 modality-dependent sensory receptors allow the brain to perceive different types of stimulus. 34 modality means energy form of stimulus. 35 coding for stimulus intensity is done in two ways, frequency coding and population coding. 36 frequency coding means the number of action potentials being generated per unit time. The number of action potentials per unit time is proportional to the strength of the stimulus. 37 population coding activates multiple free nerve endings at once. The stronger the stimuli, the more free nerve endings are activated at once. 38 first-order neuron signal sprouts to adjacent neurons to induce them to release less neurotransmitters into the synapse by presynaptic inhibition. The action potential is amplified to an exact location by the first-order neuron, known as lateral inhibition. 39 tactile acuity via two-point discrimination. The larger a tactile field, the less accurate the stimulus location is. Multiple tactile fields can perceive multiple stimulus locations with accuracy simultaneously. 40 the back has larger tactile fields than the hands or palms, less accurate tactile acuity. 41 organs of general sense are pain, temperature, pressure, and touch have common features. The receptors have simple structural design, widespread distribution like over the surface of the skin. 42 5 special sense structures are really complex and have restricted distribution, like eyesight. 43 sense of smell is olfaction, taste is gustation, hearing, balance and eyesight. 44 sense of smell stimulus is chemicals, receptor is called olfactory epithelium sending information through the first cranial nerve or the olfactory nerve. 45 gustation, sense of taste, stimulus is dissolved chemicals from food receptors are taste buds, information being sent through cranial nerve pairs 7, 9, and 10. 46 sense of hearing stimulus is sound waves, receptor is cochlea sent through nerve 8. 47 equilibrium, sense of balance, receptor vestibular complex, sent through nerve 8. 48 sense of hearing and sense of balance stimuli are mechanical, or are activated by mechanical stimuli. 49 sense of smell and sense of taste stimuli is chemical, needs to be dissolved for the receptor to accept it. 50 radiant energy is visible light, stimuli for eyesight. 51 oldest special sense is the sense of smell, is also the only special sense bypassing both thalami, goes straight to the olfactory sensory cortex, is the only sense that has a direct memory component. 52 most important special sense is vision, 40% of cerebral cortex is dedicated to integrating incoming visual stimuli. 53 general sense of pain is unique, perception of pain overrides all other sensory modalities. 54 the receptor to detect noxious stimuli allows the perception of pain, known as nociceptors or leading to pain perception. 55 thermoreceptors detect temperature, bear nerve endings in the epidermis, skeletal muscle, liver, and hypothalamus. Hypothalamus can reset the body temperature. 56 mechanoreceptors detect stretching or compression, under this are tactile receptors for touch, pressure, and vibration, baroreceptors detecting pressure and proprioceptors detecting joints and skeletal muscles. 57 chemoreceptors detecting proton concentration, partial pressure of CO2 in cerebrospinal fluid and detecting the partial pressure of oxygen and CO2 in the blood. 58 central nervous system is much more sensitive to CO2 than oxygen because CO2 is directly linked to proton concentration or pH. pH fluctuation is deadly in the body, so it's maintained vigorously. 59 spinothalamic tract is the most important pathway conveying somatosensory perception. This pathway goes to the primary somatosensory cortex, or the postcentral gyrus, where conscious awareness of a sensation begins or perception starts. 60 tract means bundles of axons physically located in the central nervous system. 61 bundles of axons running outside the CNS are called a nerve. 62 left primary sensory cortex receives information from the right side of the body and vice versa. Sensory information crosses over at the entry point or at the medulla, known as decussation. 63 temperature and pain are sent through the spinothalamic tract. 64 spinothalamic tract crosses over at the entry point, whereas the dorsal column pathway moves at the same side up until the medulla then crosses over to the other side of the brain. 65. There is somatic pain and visceral or referred pain. 66. Visceral pain comes from the visceral organs in the thoracic cage, abdominal cavity, or in the pelvic cavity. 67. The heart being compromised activates the second-order neuron, which is also connected to the left forearm and left pinky finger, numbness because of heart being compromised. 68. Elbow has a type of sensory called Ab fibers, propagates action potentials faster because of more myelination, activating an interneuron, releasing glycine-inducing IPSBs on the cell body of the second-order neuron. Significantly less APS are sent to the CNS, leading to perceiving less pain. Reason for rubbing the hurt area to relieve pain, known as gate control theory. The original signal is from a C fiber. 69. Endogenous analgesia system is the ability of the brain to block pain. The descending neuron will release neurotransmitter activating the interneuron. 70. Enkephalin is a natural opioid like morphine, however morphine is man-made. 71. Enkephalin has two functions. It reduces the amount of neurotransmitter released by the afferent neuron, hyperpolarizing the axon terminal of the first-order neuron. 72. Enkephalin also produces IPSBs at the cell body of the second-order neuron, decreasing probability of firing an action potential, ultimately decreasing pain perception. 73. Enkephalin is released by the interneuron. 74. Enkephalin has one peripheral mechanism and one central mechanism. 75. Light is radiant energy being converted by photoreceptors into graded potentials, travels back out onto the bipolar cells and the ganglion cells. The ganglion cells are the only type of neurons from the retina capable of generating action potentials, propagating APS along cranial nerve 2 or the optic nerve. 76. Primary visual cortex is located in the occipital lobe. 77. Information from the eyes goes to the thalamus, the suprachiasmatic nuclei, and to the superior colliculi. 78. Information to the superior colliculi is for the visual reflex. 79. Information to the suprachiasmatic nucleus is for the circadian rhythm. 80. Thalamus pathway goes through optic radiation and onto the primary visual cortex. 81. Phototransduction of light. 82. When there's no light or in the dark, photoreceptors are more active than in the presence of light. There's a molecule called rhodopsin in the absence of light. Cyclic GMP is not destroyed. Cyclic GMP is a secondary messenger acting as a chemical signal activating from the inside of the cell a sodium channel allowing sodium influx depolarizing the membrane, inducing calcium influx releasing neurotransmitters binding to a receptor generating graded potentials on the bipolar cell. Bipolar cell then generates more graded potentials towards the ganglion cell to fire action potentials in the dark. 83. When light strikes the rhodopsin activating the molecule, a G protein called transducin is activated. More specifically, the alpha subunit activates a transmembrane protein acting as an enzyme called phosphodiesterase. This destroys the cyclic GMP. Without cyclic GMP, the sodium channel shuts closed. The leak potassium hyperpolarizes the cell. No calcium influx, less neurotransmitter being released, less action potentials in the presence of light. Chapter 11. Somatic and autonomic nervous systems. One nervous system is under voluntary control called somatic motor division, and involuntary control called autonomic motor division. 2. Autonomic motor system also called ANS or autonomic nervous system. 3. Autonomic synonym is visceral. 4. Somatic nervous system is the somatic motor pathway, or voluntary control of the skeletal muscles. 5. Somatic motor pathways control only one type of effector organ, the skeletal muscle, by using the upper and lower motor neurons. 6. One skeletal muscle is one organ. 7. One skeletal muscle fiber is one cell. 8. Axons of upper motor neurons are bundled into tracts, like the pyramidal tract. 9. The cell body of the upper motor neuron resides in the primary motor cortex, also called the precentral gyrus. 10. Precentral gyrus is the structural name for the functional primary motor cortex, belonging to the frontal lobe. 11. Upper motor neuron sends axons down to synapse with the lower motor neuron. The cell body of the lower motor neuron is in the anterior gray horn of the spinal cord. 12. Anterior gray horn is the specific anatomical region within the spinal cord, contains all the cell bodies of lower motor neurons. 13. Lower motor neurons sense their axons out leaving the spinal cord, innervating the effector cell, in this case skeletal muscle fiber. 14. Skeletal muscle fiber is called this way because of the shape of the cell, it's a long cell looking like a fiber. 15. Cardiac muscle can also be called cardiac muscle fiber. 16. Smooth muscle cannot be called fiber, should be called smooth muscle cell. 17. Axons of upper motor neurons are called fiber tracts. 18. Axons of lower motor neurons are bundled into nerves leaving the spinal cord. 19. Axons within the brain or spinal cord are called fiber tracts. If they run outside the brain or spinal cord they are called nerves, specifically cranial nerves or spinal nerves. 20. The pyramidal tract starts out as the internal capsule, name changed to cerebral peduncle, bundles of axons belonging to the upper motor neuron running through the midbrain, going to the medulla. Starting in the medulla, the cerebral peduncle is called the pyramids, leaving and going into the spinal cord, name changed to corticospinal tract. 21. Corticospinal tract begins in the cerebral cortex, ends in the spinal cord, is a somatic motor pathway. 22. Cerebral cortex is the most intelligent part of the brain. 23. If a tract begins in the brain and ends in the spinal cord, it is a motor pathway, cannot be a sensory pathway, which starts in the spinal cord and ends in the brain. 24. Motor unit is similar to a sensory unit. Sensory unit is one single sensory neuron serving a defined number of sensory receptors. Motor unit is one single motor neuron innervating a specific number of effector cells, in this case skeletal muscle fibers. 25. Small motor units are single motor neurons innervating 2 to 3 skeletal muscle fibers, does not have small motor units innervating a single SKM fiber, at least not in humans. 26. Small motor units are for precision-guided movements. 27. Large motor units can innervate up to 4,000 skeletal muscle fibers, generating a lot of power. Disadvantage is it cannot generate precision-guided movements. 28. For every muscle as an organ there is different sizes of motor units, which are dictated by genetics. 29. Skeletal muscles display a symmetrical innervation pattern. Innervating muscles by the motor unit symmetrically, allows contraction of the entire muscle at the same time. 31. Skeletal muscle fiber is innervated by only one singular lower motor neuron. 31. When a lower motor neuron innervates skeletal muscle fibers, there's no physical contact. It is instead a neuromuscular junction. 32. Neurotransmitter in the axon terminal, acetylcholine stored in the synaptic vesicle, axon potential arrives propagated down the axon, hits the axon terminal also called terminal Bowden and dies, depolarization signal spreads to voltage-sensitive calcium channel, allowing influx of calcium channel, vesicles fuse to axon terminal, releasing neurotransmitters into the neuromuscular junction. Diffusing by simple diffusion, acetylcholine binds to the nicotinic cholinergic receptor, an ion channel. On the skeletal muscle fiber, opens fluxing 3 ionic species, sodium influx, calcium influx, and potassium efflux, net gain of positive charge, depolarizing the membrane, known as EP or end plate potential. Will always reach threshold potential on the skeletal muscle fiber. 33. Skeletal muscle fiber cell membrane is called sarcolemma. 34. EP stands for end plate potential, is always a depolarization above threshold potential. Once action potential is generated at the sarcolemma it will spread throughout the muscle fiber, contracting it. 35. Once acetylcholine is released into the neuromuscular junction, the nicotinic cholinergic receptor on the sarcolemma requires 2 acetylcholine molecules to bind to each channel before it can open. 36. Acetylcholine is neutralized by acetylcholinesterase, it will cut the acetylcholine into choline and acetic acid, the neuron picks choline up to recycle to make more acetylcholine neurotransmitter. 37. Skeletal muscles not receiving innervation from lower motor neurons stop reacting, they atrophy and die, requires neural input 24/7, only paralyzed completely during REM stage in sleep to prevent acting out during the dream. 38. Cardiac muscle and smooth muscle don't require neural innervation, they are independent and can work on their own, don't require 24/7 neural input, downside is they cannot be controlled on a voluntary basis. 39 Autonomic nervous system has three divisions: sympathetic division, parasympathetic division, and the enteric division. 40 Sympathetic and parasympathetic have opposing functions, they do not work together, they only work together during sexual climax. 41 Sympathetic division is activated under emergency situations or fight or flight situations. 42 Parasympathetic division is for rest and digest situations. 43 Most visceral effector organs receive innervation from both sympathetic and parasympathetic division, known as dual innervation. 44 Structures that receive innervation from only the sympathetic division are effector organs like smooth muscle from the walls of blood vessels, smooth muscles wrapping around hair follicles known as erector pili, as well as adipocytes, fat cells. These three are exclusively served by the sympathetic division, as well as the conduction system of the heart and the adrenal gland, and sweat glands. 45 Erector pili, when they contract they give goosebumps. 46 Cardiac muscle of the heart receives both sympathetic and parasympathetic divisions, conduction system of the heart only receives sympathetic division. 47 Conduction system of the heart gives rise to the rhythm of the heart, is the pacemaker system. They are bona fide cardiac muscle fibers, have lost the ability to contract, have gained the ability to generate spontaneous action potentials, dictate how fast and how slow the heart beats. 48 Sympathetic division can increase heart rate through the conduction system. Both sympathetic and parasympathetic can control the strength of contraction of the heart. 49 Adrenal gland above the kidney is specifically under sympathetic innervation. 50 ANS is involuntary nervous system. 51 Definition of a ganglion is an aggregation of bundles of cell bodies of neurons. The cell bodies are physically located outside of the spinal cord and the brain. Within the brain or within the spinal cord it is instead called a nucleus instead of a ganglion. 52 Motor ganglia are only autonomic ganglia. There are not somatic motor ganglia, there are somatic sensory ganglia, meaning the cell bodies of the first-order neuron located in the dorsal root ganglia. 53 Autonomic ganglion can also be called visceral ganglion. 54 Specific targets for the autonomic nervous system are cardiac muscle, smooth muscle, glands, and adipose tissue. 55 Sympathetic and parasympathetic divisions have many structural and functional differences. 56 Sympathetic division has different names. Sympathetic is a functional name, anatomical name is thoracolumbar division. This division exits between thoracic vertebrae 1 and lumbar vertebrae 2. 57 Three types of ganglia under sympathetic division with three different names. First type is sympathetic chain, second type is adrenal medullae, third type is called collateral ganglia. 58 Three collateral ganglia are located anteriorly in front of the vertebral column. 59 Sympathetic chain is on the sides of the vertebral column. 60 Names of the three single collateral ganglia are the superior mesenteric ganglion, inferior mesenteric ganglion, and the celiac ganglion. 61 Preganglionic neuron axon is myelinated, the postganglionic neuron axon is unmyelinated. 62 Chromaffin cells replace the postganglionic neuron. Because of this the adrenal medulla is called a modified ganglion, because the postganglionic neuron has been replaced by the chromaffin cells. They release chemical messengers as hormones by going into the bloodstream. 63 White ramus is axons of preganglionic neurons. These axons are white because they are myelinated. Myelination is lipid, fat-based, looks white. 64 Gray ramus is axons of postganglionic neurons. These axons are unmyelinated, they look grayish. 65 Preganglionic are considered to be central neurons. Cell body is located in the spinal cord on the lateral gray horn, between thoracic vertebrae 1 to lumbar vertebrae 2. 66 Cell body of the postganglionic neurons are located in the ganglion cells. Each ganglion contains hundreds of cell bodies belonging to postganglionic neurons. They send unmyelinated axons out to serve the effector, which is cardiac muscle, smooth muscle, glands, or adipose tissue. 67 Adrenal medullae, 80% is epinephrine, 20% is norepinephrine. They work as hormones. They can also work as a neurotransmitter depending on where they are released into. 68 Epinephrine and norepinephrine fall under another name called catecholamines. Catecholamines have adrenaline, noradrenaline, and dopamine. 69 Parasympathetic division is synonymous to craniosacral division. 70 Preganglionic neurons from brainstem in parasympathetic division, and also from the sacral spinal cord. 71 Preganglionic neuron axons are longer in the parasympathetic compared to the sympathetic division. 72 axons of postganglionic neurons are shorter from the parasympathetic compared to the sympathetic. 73 preganglionic neurons exit the spinal cord at the brainstem and at the sacral spinal cord. 74 Parasympathetic only has two types of ganglia. Ganglia in sympathetic are closer to the spinal cord, ganglia in parasympathetic are closer to the target organ. 75 Preganglionic neurons releasing ACH or acetylcholine, the neuron is then called cholinergic, this is part of the sympathetic division. 76 Postganglionic neuron releases another type of chemical messenger called epinephrine and norepinephrine, neuron is known as adrenergic. 77 The receptor is known as a nicotinic cholinergic receptor, this receptor is for ACH, this type of receptor is an ion channel, or an ionotropic or fast response receptor. 78 The epinephrine and norepinephrine receptor is called adrenergic receptor, these are metabotropic receptors or slow response receptors. 79 Table 11.1 page 311 There are different adrenergic receptor, there's the alpha and beta adrenergic receptor, for alpha there's alpha 1 and 2, there are three subclasses for beta known as beta 1, 2, and 3. 80 Parasympathetic division The same neurotransmitter ACH, preganglionic neuron releases ACH, the postganglionic neuron also releases ACH, both neurons are cholinergic, the receptor is not the same as in the sympathetic division, the receptor is a nicotinic cholinergic receptor, this is an ionotropic channel or fast response channel, the muscarinic cholinergic receptor is a G-protein coupled receptor, or metabotropic or slow response receptor. Chapter 12 Part 1, Muscle Physiology. 1 Skeletal muscle fiber means skeletal muscle cell. 2 Sliding filament model is the best model to explain muscular contraction at the molecular level, it's known as cross-bridge formation, or a fancy way of saying contraction of muscle cells at the molecular level. 3 Smooth muscle fiber contraction is slightly different to skeletal and cardiac muscle fiber. 4 The cell membrane in muscle is called sarcolemma. 5 Sarcoplasmic reticulum is the equivalent of the smooth endoplasmic reticulum in other cells, it's used as a storage and release site of calcium in muscle cells, important role of calcium to induce cross-bridge formation. 6 Within the skeletal muscle fiber is multiple types of protein, one of these structures is the myofibril, within each myofibril is the smallest structural and functional unit of muscle known as a sarcomere, each myofibril contains hundreds to thousands of sarcomeres, when the skeletal muscle fiber contracts, each sarcomere becomes shorter. 7 Each sarcomere has thin filaments and thick filaments, thin filaments are built from actin, thick filaments are made from myosin, both thin and thick filaments are proteins, interaction between actin and myosin is what gives rise to contraction, this interaction is known as cross-bridge formation. 8 Skeletal muscle fibers have many mitochondria, the entire sarcoplasm is a majority the myofibrils and mitochondria, the two predominant structures in skeletal muscle fibers. 9 Each skeletal muscle fiber contains more than one nucleus, they are known as multinucleated cells, they are sandwiched between the sarcolemma and the sarcoplasm, they are right beneath the sarcolemma. 10 When cross-bridge formation occurs, the thick filaments remain stationary, the thin filaments move toward the M-line, causing the sarcomeres to become shorter. 11 Sarcomeres are between one Z-line to the next Z-line, with an M-line in the middle that thin filaments move toward. 12 Cross-bridge formation is the cellular level name for contraction, myosin interacts with actin causing the thin filaments to move towards the sarcomere M-line. 13 I-band has only thin filaments, looks lighter, A-band has both thick and thin filaments overlapping, looks darker. 14 Cross-bridge formation is the interaction between myosin from the thick filament with the actin on the thin filament. 15 At rest when calcium is not present, each actin protein on the thin filament has a myosin binding site. At rest the myosin binding site is occupied by a protein filament called tropomyosin, a filamentous protein, blocking myosin from binding to actin, requires calcium to interact with troponin complex, Troponin complex induces a structural change, pulling tropomyosin away from the actin binding sites, freeing up the actin receptors for myosin. 16. Two filaments of actin interacting to make a thin filament is called a double helical actin strand. 17. The head of myosin has two components, an actin binding site and an enzyme capability or ATPase site. 18. The myosin ATPase has different rates, the fastest one can cycle up to 5 cross-bridge formations per second. 19. ATPase means it hydrolyzes ADP to harvest energy. 20. Cross-bridge formation requires calcium and ADP, calcium allows the interaction of myosin and actin, ADP disengages myosin from actin. 21. Rigor mortis, or the stiffening of the body after death, is caused by myosin not disengaging from actin because of a lack of ADP. 22. Myosin cannot disengage from actin without ADP, myosin head has a higher affinity for ADP than for actin, it instead binds to ADP, hydrolysing it, attaches to the next actin. 23. Efficiency is provided by out of phase between cross-bridge formations, uses same energy yet generates a longer duration for cross-bridge formation. 24. Motor end plate is the region on the sarcolemma that is on the opposite side of the axon terminal, this has a high density of ACH receptors, on the other side of the neuromuscular junction. 25. The invagination of the sarcolemma going inside the cell is called the T-tubule. 26. The sarcolemma along the T-tubule sits next to the sarcoplasmic reticulum. There are transmembrane proteins allowing the sarcolemma to detect incoming action potentials on the sarcolemma of the T-tubule, recognizes the signal and releases calcium into the sarcoplasm. 27. End plate potential is acting like a spark plug, to induce the muscle to generate its own action potential, it ignites the generation of action potentials on sarcolemma, because it has its own voltage-sensitive sodium and potassium channels. 28. When action potential is spreading on sarcolemma and goes into the T-tubule, there are two transmembrane proteins, on the membrane of the sarcolemma T-tubule it contains the DHP receptor, on the membrane of the sarcoplasmic reticulum is the ryanodine receptor. 29. Action potential on the T-tubule induces a shape change or conformation of the DHP receptor, also induces a change on the ryanodine receptor, it's like opening a lid of a can, the calcium goes into the sarcoplasm, instead of staying in the sarcoplasmic reticulum. 30. Every time an action potential propagates throughout a skeletal muscle fiber sarcolemma and the T-tubule, it will be detected by transmembrane proteins sensitive to the arrival of the depolarization signal, induces the DHP receptor to change shape, also causes a shape change on another interacting protein called a ryanodine receptor located on the membrane of the sarcoplasmic reticulum. The shape change allows an opening to influx calcium, an example of facilitated diffusion, calcium interacts with troponin. Chapter 12 Part 2, Muscle Physiology Part 2. 1. Force or tension generated by individual muscle fibers, affected by frequency of stimulation. 2. There's a lag or latent period before the muscle responds to the stimulus. 3. Twitch in a muscle is one contraction, or thousands of cross-bridge formations within one skeletal muscle fiber contracting each a singular time. 4. Stimulus for muscles is a depolarization signal, it's an excitation-contraction coupling. 5. The lag called the latent period is caused by the time it takes for the stimulus to reach the entire skeletal muscle. 6. During the contraction phase the action potential is still spreading throughout the sarcolemma of the skeletal muscle, as soon as it dissipates it leads to a relaxation phase. 7. Sarcoplasmic reticulum reclaims calcium as soon as the relaxation phase is over. 8. Isometric twitch or isotonic twitch. Isometric means the muscle is contracting without changing the length of the muscle, isotonic twitch means the length of the skeletal muscle decreases during contraction. 9. Isometric twitch is when the muscle cannot be moved by the skeletal muscles, it tries to contract but it cannot. Isotonic twitch means the skeletal muscles are moving and contracting, and lifting whatever it is with it. 10. TREP is an observation, the exact molecular mechanism is still not clear to science. There's a capability in skeletal muscle tissue that as the same stimuli is exposed to the skeletal muscle cell, the tension increases until it plateaus at the fourth application of the same stimulus. 11. When the muscle contracts they can be added together, this is called summation. The next level is incomplete tetanus, gives really shaky movement, Complete tetanus means the contractions are so close in time they effectively become fused and gives rise to smooth body movements. 12. Force generated by individual fibers is impacted by the diameter of each individual muscle fiber dictates the tension that can be generated. The larger the diameter, the stronger the muscle fiber. 13. The sarcoplasm contains much more filaments in larger muscle fibers for cross-bridge formation capability, more contraction, and more tension. The cell generates much more forceful contraction. 14. Skeletal muscle fiber force generation is affected by changes in fiber length. Optimal length for maximal force generation is 100 to 120% of resting skeletal muscle fiber length. 15. Contractile cardiac muscle at rest is not at the optimal force generating length. When they are stretched out, then they can generate maximal force. Serves the purpose of incoming blood stretching the heart wall. Stretching allows the cardiac muscle fiber to be at its strongest optimal length for contraction, efficiently pumping blood out of the heart. 16. Three factors can affect the tension generated by entire skeletal muscle organs. 17. Recruitment refers to how many motor units have been activated in a skeletal muscle organ, affects the strength of the skeletal muscle organ. Recruitment means increasing the number of active motor units. 18. Smaller motor units are recruited first. If more power is needed, larger motor units are recruited. Larger motor units innervate more skeletal muscle fibers. 19. Force generated by whole muscles is also affected by size principles. Both the size of motor units and the motor neurons controlling motor units also vary in size. 20. Stronger synaptic input, via spatial and temporal summation, is required to induce larger neurons to fire. 21. The skeletal muscle fibers in larger motor units are also larger themselves. They have larger diameters as well. The larger the motor unit, the more skeletal muscle fibers innervate, and the larger in diameter those skeletal muscle fibers are as well. 22. There are different types of skeletal muscle fibers. They have different speeds at which they can contract, fast twitch fibers versus slow twitch fibers. 23. All skeletal muscle fibers have the same latent period. The fastest are extraocular muscles, 6 of them, inserting into the eyeball, 7 to 8 milliseconds time to reach maximal tension. 24. Gastrocnemius muscles, calf muscles in the leg, take 40 milliseconds to reach maximal tension. 25. Soleus, deep to gastrocnemius, muscles takes 90 milliseconds to reach maximal tension. 26. Slow oxidative or red fiber versus the fast glycolytic or white fiber. 27. Glycolytic or white fiber does not require oxygen. It utilizes glycolysis. No oxygen means the mitochondrial density is low, as well as blood vessels perfusing the muscle is not required, nor is myoglobin storing oxygen in muscle required. 28. Glycolytic or white fibers fatigue very fast. Diameter size and motor unit size are large and generates powerful tension. 29. Slow twitch or red muscle fibers require oxygen, needs a lot of mitochondria, capillary density, myoglobin to store oxygen, resists fatigue well, smaller in motor unit size and diameter of muscle fiber and weaker tension capability, low myosin activity leading to slow speed of contraction. 30. Extrafusal fibers are contractile muscles innervated by alpha motor neurons or the lower motor neurons. Another type of lower motor neurons are gamma lower motor neurons. Both the gamma and the alpha are synapsing with the upper motor neuron. 31. Alpha motor neurons have a bigger diameter than gamma motor neurons. They propagate the action potentials faster. The extrafusal fibers always contract first. 32. Gamma motor neurons do not innervate extrafusal fibers. They innervate intrafusal fibers, which do not contract the muscle as a whole. They contract in such a way to reflect the contraction of the entire organ, sends the information back to the CNS. 33. Muscle spindles detect the change in length of the entire organ, known as intrafusal fibers. 34. The length decrease of the skeletal muscle needs to be sent to the CNS by the intrafusal fibers or the muscle spindles. 35. The intrafusal fibers contract slightly later to match the extrafusal fibers contracting. 36. Sensory afferents detect the change in length of skeletal muscle fibers by the intrafusal fibers. 37. Extrafusal fibers are not part of the muscle spindle. They do not relay information back to the CNS about the length status of the entire organ. 38. Tendons are part of the skeletal muscle, connecting it to the bone. 39 Golgi tendon organs inhibit a reflex. As the muscle is stretching out, the reflex is to contract. Under some circumstances the muscle is stretching forcefully enough that contracting would tear the tissue. Inhibiting the reflex saves the tissue. 40 Tendons cannot get ripped off the bone is most likely to break instead. Chapter 13, Cardiovascular System, Cardiac Function. 1. Cardio means the heart, vascular means the blood vessels. 2. Cardiovascular systems consists of the heart, blood vessels, and the blood itself. 3. The heart has two components, an electrical component and a mechanical component, this is the cardiac function. 4. Cardiac cycle refers to the mechanical component of the heart. 5. Electrical component of cardiac function dictates the rate of the heart. 6. The heart does not require neural input, or decisions from the central nervous system is not required to function. The heart is independent of the central nervous system. 7. Skeletal muscles require neural input constantly, the cardiac muscle does not need neural input to function. 8. The central nervous system does influence cardiac muscle function, but is not required for the function of cardiac muscle. 9. The mechanical cycle of the heart generates the pressure to push blood through the entire vascular bed. 10. The amount of blood ejected by the heart is called cardiac output. 11. There are two types of cardiac muscle, there are pacemaker cells and cardiac contractile cells, both are two types of cardiac muscle cells. 12. Pacemaker cells are cardiac muscle cells, they have lost the ability to contract, to generate cross-bridge formation, and have gained the ability to generate action potentials, these action potentials will dictate the rhythm of the heart. 13. Cardiomyocytes are referring to the cardiac contractile cells. The pacemaker cells in anatomy are part of the conduction system. 14. The myocardium contains the cardiac muscle fibers that generate cross-bridge formation to contract the heart. 15. In neurons, depolarization is carried by a massive influx of sodium cations into the neuron. In the heart the depolarization is similar, carried by a sodium influx. 16. Rapid depolarization in cardiac pacemaker cells is not mediated by a massive sodium influx. 17. The hyperpolarization potential in cardiac pacemaker cells opens new types of ion channels. 18. Cardiac pacemaker potential is a spontaneous potential, or a generated spontaneous depolarization. 19. Cardiac pacemaker potential has two parts to it, mediated by two different ionic species. 20. Page 371 Figure 13.12 in textbook and page 372 Table 13.1. 21. The first phase of the cardiac pacemaker potential is due to sodium influx and potassium efflux through channels called funny channels. They open in hyperpolarization territory. There's much more sodium influxing than potassium effluxing, depolarizing toward threshold potential. Funny channel can only get from -70 to -55 millivolts, fails to get to threshold potential, but this depolarization opens another ion channel. 22. The depolarization from the funny channels opens another ion channel that causes a massive calcium influx into the cardiac pacemaker cell. The ion channel is called the T-type calcium channels. The T stands for transient, they open only briefly. 23. The T-type calcium channels allows the cardiac pacemaker cells to reach threshold potential to fire action potentials. 24. The massive depolarization of the cardiac pacemaker cell in an action potential is caused by a massive calcium influx, through a different ion channel called an L-type calcium channel. The L stands for long-lasting, meaning they stay open for longer to allow calcium to flux in to depolarize the cell membrane of the cardiac pacemaker cells. 25. The repolarization of the cardiac pacemaker cells in action potentials is similar to neuron repolarization. 26. Resting membrane potential in cardiac pacemaker cells is unstable and fluctuates. Reaching resting membrane potential activates the funny channels, depolarizing the cell. 27. Spontaneously the cardiac pacemaker cells can generate action potentials on their own without the intervention of the central nervous system. 28. Cardiomyocytes generate cross-bridge formation to contract in order to push blood out of the heart. 29. Figure 13.13 page 372 goes with table 13.2 page 373. 30. There are 5 phases in an action potential for a cardiac contractile cell or a cardiomyocyte. 31. Cardiomyocytes have a stable resting membrane potential unlike cardiac pacemaker cells. 32. Page 372 to 373 reading. 33. Phase 0 in an action potential in cardiomyocytes is caused by a massive sodium influx, similar to neurons. 34. Phase 2 maintaining depolarization is due to calcium influx, through the L-type calcium channels. 35. Cardiac contractile cells have 2 types of potassium channels. During phase 1 the potassium channels are called inward rectifier channels. This is a type of potassium channel, closed during phase 1, it's also a voltage-sensitive potassium channel. 36. The plateau in cardiac contractile cell action potentials is mediated by 2 components, the closure of the inward rectifier channels and the opening of the L-type calcium channels. 37. Repolarization is caused by the opening of another type of channel called delayed rectifier channels. They open to allow potassium efflux to repolarize during phase 3. During the repolarization the inward rectifier channels are also open, both of them mediate the potassium efflux. 38. Phase 4 is the resting membrane potential. 39. Cardiac contractile muscle cell resting membrane potential is at -90 mV. 40. Cardiac pacemaker cell resting membrane potential is at -70 mV. 41. The action potential travels between neighboring cells through gap junctions. Innervating a single cardiac contractile cell can depolarize the entire heart through gap junctions. 42. Every single skeletal muscle fiber requires a neuromuscular junction with the lower motor neuron axon terminal to contract. In cardiac muscle fiber there's no requirement of this. Exciting one single cardiomyocyte excites the entire heart through gap junctions. 43. Cardiac cycle refers to the mechanical component of the heart, pumping of the heart. 44. EKG refers to the electrical/conduction component of the heart, rhythm of the heart. 45. In the clinical setting, there's no way to monitor the mechanical component of the heart without being invasive. Following the EKG of the heart can roughly follow the mechanical component of the heart. 46. EKG can also be ECG. 47. Electrical component of the heart can be picked up by electrodes placed on the skin. 48. 1 EKG is electrical activity for the entire heart. 49. The P part of the EKG is called P deflection. This is caused by atrial depolarization, both atria are being depolarized. 50. Halfway through the P deflection is atrial systole. 51. Systole means contraction. 52. Atrial systole is the mechanical component. When both atria are contracting, only contract after being depolarized. 53. From the EKG you can infer the cardiac cycle. 54. Following the P deflection is the QRS complex, a massive signal. 55. The QRS complex includes 2 events, includes the atrial repolarization and ventricular depolarization. 56. The QRS complex is caused by one larger ventricular depolarization and one smaller atrial repolarization. 57. During the QRS complex it can be inferred the atria are relaxed and the ventricles are contracting. 58. Following atrial repolarization, the atria are in diastole, or both atria are relaxed. 59. Diastole means relaxation. 60. The ventricles right after depolarization are in ventricular systole. 61. After ventricular systole or depolarization the ventricles are in ventricular repolarization, this is reflected by the T in the electrocardiogram chart. 62. Following ventricular repolarization is ventricular diastole, the ventricles are relaxing. 63. The QT interval is the duration of ventricular systole, or when the ventricles are contracting. 64. The TQ segment is when both ventricles are relaxed or not contracting, called ventricular diastole. 65. The RR interval is the time between each heartbeat. 66. The PQ interval is the time when both atria are in systole, or when both atria are contracting. 67. Isovolumetric contraction and isovolumetric relaxation, during both of these all 4 cardiac valves are closed. 68. Isovolumetric contraction means the ventricles are starting to contract and squeeze, the pressure is strong enough to close the right and left atrioventricular valve, not strong enough to open the semilunar valves. 69 Isovolumetric relaxation is when the ventricles start to relax, the pressure inside the ventricles drops, still too high to open the atrioventricular valve, the pressure is less than the pressure in the ascending aorta and the pulmonary trunk, shuts closed the semilunar valves as well. 70 Lub DUPP refers to heart sounds. Lub is a lower sound due to the closure of the atrioventricular valves, DUPP is a higher sound due to the closure of the semilunar valves. The valves closing does not make a noise, the sound heard is due to blood turbulence, the blood flows back into the valves and makes a noise. 71 Cardiac output is the amount of blood being ejected from the heart. 72 Cardiac output equals heart rate, multiplied by stroke volume. 73 Heart rate is the electrical component of the heart. 74 Stroke volume is the mechanical component of the heart. 75 Heart rate is controlled by extrinsic factors, or factors coming from outside the cardiovascular system. 76 The sympathetic division of the autonomic nervous system releases norepinephrine or epinephrine. Norepinephrine is released acting as a neurotransmitter, released by the postganglionic neuron of the sympathetic division into a neuromuscular junction. 77 Epinephrine is also released by the autonomic nervous system, but is released as a hormone into the bloodstream by the adrenal medulla, specifically by the chromaffin cells within the adrenal medulla. These are modified ganglia. 78 Norepinephrine and epinephrine bind to the same receptor in the pacemaker cells. They bind to the beta-1 G protein coupled receptor. The receptor activates the G protein called GS, or the alpha subunit, recognizes and binds to the adenylate cyclase membrane-bound enzyme. The adenylate cyclase converts ADP into cyclic AMP activating protein kinase, an enzyme capable of adding phosphate groups onto other targets, activates funny channels and transient-type calcium channels depolarizing the membrane, allowing the pacemaker cell to generate more action potentials per unit time. The outcome is the heart rate increases, because of the increase in action potentials. 79 The parasympathetic division of the autonomic nervous system uses acetylcholine as a neurotransmitter, which is released by the postganglionic neurons from the parasympathetic division of the autonomic nervous system. Heart rate is still controlled by extrinsic control. 80 Acetylcholine binds to the receptor called muscarinic cholinergic receptor, G protein coupled receptor, activating the alpha subunits, opening a potassium channel. Potassium effluxes, hyperpolarizes the cell membrane of the pacemaker cell, also closes T-type calcium channels, lowering the amount of action potentials being produced, lowering the heart rate. 81 Sympathetic system decreases the heart rate, the parasympathetic system increases the heart rate. 82 The parasympathetic division cannot influence how strong or how weak the heart contracts, it can only influence the electrical component, or the rhythm of the heart. 83 Only the sympathetic division can increase the strength of contraction of the heart. 84 Protein kinase induces adipostate to pump in calcium. 85 End diastolic volume affects stroke volume, is the amount of blood inside each ventricle at the end of the ventricle relaxation phase. 86 Cardiac muscle at rest is not at maximum length, blood entering the heart stretches the muscle, inducing the sarcomere into an optimal position for the maximum strength contraction. 87 The amount of blood coming back to fill the heart is known as end diastolic volume. 88 End diastolic pressure is also preload, or the greater time between beats allows more blood into the heart. 89 Atrial pressure depends on venous return. 90 Afterload affects only individuals under pathological conditions, the amount of pressure the heart needs to overcome before blood can be pumped into the aorta and the pulmonary trunk. 80 mmHg for the aorta and 8 mmHg for the pulmonary trunk. 91 The top number is referred to as systolic pressure, the blood pressure when the ventricles are contracting. The bottom number is systolic pressure, or when the ventricles are relaxing. The indicator of long-term health is the bottom number. The higher the systolic pressure, the more pressure needs to be generated by the left ventricle to open the aortic semilunar valve, stressing the heart for each contraction. Chapter 14, CVS blood flow and blood pressure. 1 CVS stands for cardiovascular system. 2 Intrinsic control mechanisms of blood flow, inside the cardiovascular system. 3 Extrinsic control mechanisms of blood flow, outside of the cardiovascular system. 4. Regulation of mean arterial pressure, extremely important. 5. Blood flow being controlled by the blood vessels themselves. There are 4 intrinsic mechanisms. First is called active hyperemia. 6. Hyperemia means anything pertaining to an increase in blood perfusion through an organ, or an increase in blood flow. 7. At rest, tissues will consume the same amount of oxygen and nutrients being produced by the blood vessels. The disposal of the cellular waste coming from the cell being picked up by the blood is also equal. Under these conditions, blood flow and blood vessels stay the same. 8. Intrinsic control of blood flow has 4 types: active hyperemia, reactive hyperemia, myogenic response, and regulation by locally secreted chemical messengers. 9. Increase in metabolic activity means the tissues require more oxygen and nutrients, and have more waste that needs to be picked up. The blood vessels detect this change and respond. The blood vessels expand and increase blood flow, allowing more oxygen and nutrients being brought in, and more cellular waste can exit by the blood vessels. 10. Reactive hyperemia is caused by blood flow suddenly dropping. Most common cause is a blood clot. Reaction is similar to active hyperemia. The blood vessels dilate to allow more blood to come in. 11. The wall of blood vessels has smooth muscle. The walls of the heart have cardiac muscle. Both when stretched react by contracting, known as vasoconstriction in blood vessels. The diameter of blood vessels becomes smaller. 12. When the smooth muscle wall in blood vessels will be stretched out, it will constrict, known as a myogenic response. 13. The release of chemical messengers is another intrinsic control mechanism of blood flow. 14. The most powerful vasoconstrictor naturally produced by the body is not included in the list, called angiotensin II, a chemical released by another organ system, so it's extrinsic instead of intrinsic. 15. The smaller the diameter of a tube, the higher the resistance of the liquid flowing through that tube. 16. The difference between the pressures of areas allows liquids to flow from the higher pressure area to the lower pressure area. 17. Flow in human physiology is known as cardiac output, the amount of blood pumped out by the heart per unit time. 18. Cardiac output depends on the heart rate multiplied by the stroke volume. 19. Total peripheral resistance is TPR. 20. Cardiac output is equal to mean arterial pressure divided by total peripheral resistance. 21. Mean arterial pressure is equal to cardiac output times total peripheral resistance. 22. Mean arterial pressure is equal to stroke volume times heart rate times total peripheral resistance. 23. The force driving blood forward depends on stroke volume, heart rate, and total peripheral resistance. 24. Total peripheral resistance comes from the vascular beds. 25. Norepinephrine is coming from the nervous system. 26. Epinephrine is from a sympathetic response, from the autonomic nervous system, however it is released by the medulla of each adrenal gland. Epinephrine is released into the bloodstream. Under this context, epinephrine is not acting as a neurotransmitter, even though the release was induced by the nervous system, it's acting as a hormone. 27. Norepinephrine is released as a neurotransmitter, released by postganglionic fibers, released by axon terminals belonging to postganglionic neurons. 28. Alpha-1, beta-1, and 2 are all G-protein coupled receptors. 29. Mean arterial pressure drives blood through the blood vessels. 30. By the time the blood gets back to the right atrium, mean arterial pressure is exactly 0. When it comes out of the right aorta, it's about 120 mmHg. 31. The mechanical component of the heart pumps the blood without mean arterial pressure, instead it generates the pressure itself. 32. Any factor that affects heart rate, stroke volume, and total peripheral resistance will also affect mean arterial pressure. 33. The only 4 blood vessels perfusing the brain are the right and left carotid arteries and the vertebral arteries. 34. The pressure in the blood vessel increasing expands the wall of the blood vessel. That mechanical action of expansion activates the neurons, inducing them to increase the frequency of the action potentials being produced per unit time. This is how blood pressure is maintained by the nervous system. 35. Information coming from the heart is going through the cardiovascular center in the brainstem, specifically the medulla oblongata, monitoring cardiovascular health constantly. Receiving information, it sends out visceral autonomic motor commands, or not consciously aware of these commands. 36. Cerebral cortex is the seat of consciousness. 37 Parasympathetic division is the rest and digest division of the visceral and autonomic motor system, mobilized in the relaxed state, sends postganglionic fibers to make neuromuscular junction with the SA node of the heart, or the electrical component of the heart. 38 The parasympathetic division cannot affect the mechanical component of the heart, it does not make neuromuscular contact with the myocardium, or the cardiac muscle cells that contract, it cannot control them without contact. 39 The sympathetic division readies the body to face emergency situations, affects both the electrical component, heart rate, and the mechanical component, stroke volume, of the heart, targets the SA node, as well as sending down axons forming neuromuscular junctions with the myocardium. 40 The sympathetic division can affect the vascular beds, it can constrict the blood vessels depending on the location or type of tissue, it can also dilate the blood vessels. 41 Arterial baroreceptors detect arterial pressure drops. 42 Arterial baroreceptors increase sympathetic division and decreases parasympathetic division. 43 Mean arterial pressure decreasing is picked up by the baroreceptors known as baroreceptor reflex. Parasympathetic activity will decrease, sympathetic activity will increase, causing the heart to beat faster, and the myocardium to contract stronger, increasing mean arterial pressure in response. 44 Stroke volume equals end diastolic volume and systolic volume. 45 End diastolic volume depends on venous return, the more venous return the more diastolic volume. 46 The sympathetic division can affect arterioles by causing restriction, increasing resistance to flow, which increases mean arterial pressure. 47 The re-establishment of mean arterial pressure is done by the sympathetic system. 48 The nervous system can affect mean arterial pressure. 49 Angiotensin II is the most powerful vasoconstrictor. 50 Epinephrine is released by the adrenal medulla, produced and released by chromaffin cells, released into the bloodstream and affects many systemic tissues, activates beta-1 in cardiac tissue, increases the heart rate and stroke volume. 51 Epinephrine can also activate beta-2 receptors. They are located in the smooth muscle on the walls of blood vessels perfusing skeletal muscle and cardiac muscle. The blood vessels dilate and allow more blood to go into skeletal and cardiac muscles. 52 Epinephrine can activate alpha-1 receptor, only if the concentration of epinephrine is high. Alpha-1 receptors do not have high affinity for epinephrine, they are located at the cell membrane of smooth muscle cells in the wall of blood vessels perfusing systemic tissues, except for cardiac and skeletal muscles. 53 Activation of alpha-1 receptors results in vasoconstriction, excludes skeletal and cardiac muscles. 54 Norepinephrine released from the nervous system activates alpha-1 receptor, causing vasoconstriction of blood vessels excluding skeletal and cardiac muscles. 55 Epinephrine acting on beta-1 receptors on the cell membrane of the pacemaker cell increases heart rate. On the contractile cell it increases the stroke volume. 56 Epinephrine acting on beta-2 receptors on the cell membrane of smooth muscle cells in the walls of blood vessels perfusing skeletal and cardiac muscles. In these cases epinephrine causes these blood vessels to dilate, increasing blood perfusion.